All right, everyone, welcome to the third set of notes for Chapter 5. These are on diffusion. So the standard here is comparing passive and active transport again. So we've mentioned this before. There are three types of passive transport, and these definitions are a little bit different than the ones that I gave in the beginning in the first set of notes. These are a little bit more detailed. So diffusion, it's the movement of molecules or solutes from an area of high concentration to an area of lower concentration. Osmosis is the same thing. It's the movement of water molecules, which are often the solvent in a solution, from an area of high concentration to an area of low concentration. Facilitated diffusion is the same, high concentration to low concentration, except for they need the transport proteins in order to cross the membrane. Notice that the common feature for every type of passive transport is from an area of high concentration to an area of lower concentration. Area of high concentration to an area of lower concentration. Again, high concentration to low concentration. This is going to be very important. It's how you actually identify something as passive or active transport. Because in order to go from an area of low concentration to high concentration takes energy on the part of the cell. So if you see the tagline, high concentration to low concentration, it will be passive transport, and it will be one of these three. So let's take a look at what we mean by molecules moving from an area of high concentration to an area of low concentration. The first thing that you have to understand when talking about a concentration is that it's a gradient. We're comparing one thing to another. So in this case, this is gray, but is it light or is it dark? Well, that depends on what you're comparing it to. This middle shade of gray is dark compared to the right-hand side over here. This shade of gray is light compared to this side. So when we say higher concentration versus lower concentration, you have to have that comparison component. And that's sort of what a gradient is. It's a scale with measurable values that can change for a given variable. For example, a thermometer is a type of gradient for the amount of heat. The pH scale is a gradient based on how basic or acidic something is. And even with light, you can measure light in foot candles from very dark to very bright. So a gradient is simply this right here. It's a scale with measurable values that goes from one extreme to the other. So you can describe this as high concentration and this is low concentration, or you can describe this as high concentration and this is low concentration. Either way. So a concentration is actually the amount of substance in a given volume. So let's go back to middle school when you were studying solutions. You need two parts to determine concentration. You need your solvent and you need your solute. And if you can pull out of your memory banks from middle school, when you talked about solutions, it has two components. The solvent is the substance that's in the greatest volume, and it does the dissolving. The solute is the substance of the smaller volume, and it's the thing that's being dissolved. So let's take a look at this example of salt water. Here we have the solvent, which in our case would be what? The thing that's in the greatest volume. In this case, it's water. And the solute is the thing that's in the smallest volume, and in this case, it would be the salt. So you put the salt in the water, the solvent, and it dissolves, which means it spreads out evenly within the solvent. Okay, so let's take a look at this from the perspective of diffusion. We said the definition of diffusion was particles moving from an area of high concentration to an area of low concentration. So if you look at the orange little molecules here, and let's pretend they're salt. You have a high concentration of salt right here. It's all located in the same area. You have a very low concentration of salt down here. So as diffusion takes over, these particles are going to move from a high concentration right here, and they're going to start to move to a lower concentration, which would be here, further down in this beaker of water. In the end, an equilibrium is reached, where there is basically about the same amount of space between each of the particles. So, passive transport is when we move from a high concentration to a low concentration. So the key thing here is, why do things move from a high concentration to a low concentration? Think about this graph as a cell starving for oxygen. So let's say this is the inside of the cell, the intracellular space. This is the outside of the cell, the extracellular space. And let's say these blue pentagons are, actually those are hexagons. So these blue hexagons, let's say that they are oxygen molecules, and our cell here is in desperate need of oxygen so that it can break down glucose and make ATP. Why is this oxygen going to move into the cell? And the reason for that, there's a couple of reasons. The most important, and the thing that we're going to focus on, is the fact that molecules are constantly moving, especially within a fluid environment. And there is fluid surrounding our cells. The cytoplasm is a fluid inside our cells. So these molecules are in constant motion. The idea that all movement would stop is a theoretical idea, actually, because atoms are constantly moving and the particles are moving. So as these particles move around in their space, they start to bump into one another. Think of them as ping pong balls. So you have all these ping pong balls in this space, and they are bouncing off of each other. Eventually they're going to bounce into the phospholipid bilayer, and some of them will actually cross the membrane. As they continue bouncing around, the likelihood that they are going to spread out increases as they continue to bounce off of each other. This is an equilibrium, where there's about the same amount of collisions occurring between the oxygen molecules on the outside of the cell and the inside of the cell. So these molecules are simply colliding with each other, and because they're concentrated, because there's a lot of them, the collisions occur more frequently, and every time the molecules hit each other, they bounce off and they're sent flying off in another direction. So the more particles there are in one space, the more likely that they are going to bounce off of each other and fly out in all directions. So I'll give you a moment to just look at that. You can pause it and write it down if you'd like. I'm going to give you one more example, one that you probably won't ever forget. So this has happened to all of you, I'm sure. You're sitting in class, minding your own business, maybe even taking notes or reading a book, and you look around and what is that horrible smell? Someone near you has farted. Now, if you think about this from a diffusion standpoint, those molecules are highly concentrated right next to the perpetrator's rear end. Yes? Okay, so as the person farts, and there's a high concentration of those particles that smell right near their rear end, it inevitably spreads, right? Depending on how bad it is, it spreads to the entire room. You've got diffusion going on. A high concentration, the moment they farted, those particles are going to move to where there is a lower concentration because they are constantly colliding and being sent off in a different direction. Unfortunately, the direction that they're sent off in happens to be near your nose. So, you look up when you notice this smell, and it seems like you're the only one who smells it. And you're looking around, and you're thinking, oh my gosh, this is awful, I'm going to throw up in my mouth. And then you notice people around you start noticing it too. It is spreading out. It is going to fill up all the available space in the room. Now here's my question. After a little while, you don't smell it anymore, right? Did those particles just evaporate? Did they just disappear into thin air? Oh no, they're still there. It just so happens that they've spread out enough that they don't trigger a smell response when they hit your nose. It's kind of gross to think about, huh? But they don't ever actually go away. They just disperse or diffuse enough that your nose can't detect them anymore because the concentration isn't high enough. So that's diffusion. High concentration near the perpetrator's rear end to low concentration, which happens to be near you or wherever you're sitting. That's diffusion. High concentration to low concentration. And it is passive transport. It doesn't require any energy. The energy of movement comes from the collisions of these particles slamming into each other and then bouncing off in opposite directions. So eventually, an equilibrium is reached. One thing to know about an equilibrium is, so here's a simulated membrane. Okay, and a simulated membrane has holes in it so that things can pass through. We know that our cell membranes are much more complicated than that. So they have a water and they put a porous membrane, or a membrane with holes in it, in the water, and they add some dye. As the dye particles bounce off of each other, probability states that over time, half of the molecules will end up on this side of the membrane and half of the molecules will end up on this side. So as they collide and bounce, some of them actually make it through the holes in the membrane. Given enough time, eventually there would be an average of 50% of the molecules over here and 50% over here. It would be an average because these molecules, once they reach this equilibrium, they don't stop moving. These dye particles aren't suddenly going to clap their hands and say, "Stand still." They continue to move. And as they collide, the probability that one of these molecules is going to cross the membrane this way is equal to the probability that as these molecules bounce off of each other, another one will move this way onto this side. So at any given moment, if you stop it and count the number of molecules on the left and the molecules on the right, it may not always be exactly equal, but over time, it will average out to about 50-50. In this case, diffusion goes both ways. So if you have some green dye particles over here and some blue ones over here, you're going to get the green ones colliding with each other and slowly moving to this side. You're going to get blue ones colliding with each other and moving to this side. But if you notice, you have a high concentration of blue molecules and a lower concentration of green. The blue is probably going to cross the membrane at a faster rate. So in our second picture, we have 3 blue ones and 1 green one. This is because the more molecules there are, the probability of them bumping into each other and knocking one across the membrane increases. Because there are fewer molecules on the right-hand side, the likelihood or the probability that they will bump into each other and knock one to the other side of the membrane is lower. But given enough time, at enough collisions, there will be an equilibrium of molecules on both sides of the membrane. So that's what we mean by equilibrium. It's dynamic, which means they don't ever really stop moving. There's just an average of 50-50 over time. So, how does this affect cells? Diffusion involves solutes. So gas particles such as oxygen and carbon dioxide, they can slip right through those cell membrane, the phospholipids of the cell membrane, without using any energy. So think about this, the oxygen molecules. The oxygen molecules get used and consumed in the mitochondria in order to make ATP. A byproduct of making ATP is carbon dioxide. Well, let's say you're making lots of ATP. You're using up lots of oxygen in the cell. So the concentration of oxygen molecules inside the cell decreases, which means there will be a higher concentration of oxygen molecules outside the cell when your mitochondria is running full steam and making lots of ATP. Because of the collisions of those oxygen molecules with each other, the probability is very high that they will bounce into the cell. Now, carbon dioxide, on the other hand, as the mitochondria makes lots of ATP and uses up lots of oxygen, it puts out lots of carbon dioxide. And the carbon dioxide builds up on the inside of the cell. Well, as it builds up on the inside of the cell, suddenly you have a high concentration of carbon dioxide inside the cell and a low concentration of carbon dioxide outside the cell. So the carbon dioxide starts to, through those random collisions and probability, the carbon dioxide starts to move out of the cell. Kind of interesting, huh? Or maybe not. So that's diffusion. Diffusion deals specifically with solutes. Oxygen and carbon dioxide are the ones that we'll focus on the most. Osmosis, on the other hand, deals specifically with water. The same thing happens with water. It moves from a high concentration to a low concentration. Cells constantly deal with water moving freely in and out of their cell. Now, this can be a good thing or it can be a bad thing, depending on their environment. And that's the next set of notes, the osmosis in action, notes 4. So right now, we're just talking about the idea of moving from high concentration to low concentration. So the last one is facilitated diffusion. In our notes the other day, we talked about how particles had to have the right size and the right chemistry in order to slip through those phospholipids. And the majority of molecules can't just slip through. They have to have help through the form of carrier proteins. Now, here's a protein channel. It looks like a straw. Again, this is specific for a certain type of molecule. In this case, it's the little round green ones. Here's a different type of carrier protein. There's no ATP listed here, so this is a passive transport type of protein, a carrier protein. And if you see it's specific to this shaped little molecule here, and it carries membranes across, or carries particles across the membrane. These also work on the idea of high concentration to low concentration, because if there is a high concentration of these little round green ones on the outside of the cell, then the probability is high that they will be bouncing off of each other and other molecules, and that they will land here and get pulled through the channel protein. The same thing is true for these oval-shaped ones that are a little bit more teal in color. So the more molecules there are outside the cell, the probability increases that as these bump into each other, that one will actually land in the protein and be carried across the membrane. So it all has to do with molecules randomly bumping into each other and the probability being high when there's a high concentration that that molecule will pass through the membrane. And that's it. I have a short video on facilitated diffusion. So I'm going to put this in here. Notice the particles are still moving. Dynamic equilibrium. The molecules are still moving. And you notice they're going both ways. right. Let's go back to our notes. So that's facilitated diffusion, and I like that little animation. I hope you enjoyed it. All right, so passive transport, quick review. This would be an exit ticket in class. Diffusion, small solutes like oxygen and carbon dioxide. Osmosis deals specifically with water. Facilitated diffusion deals with larger particles and ions. And remember that the ions are those particles that have a charge, either positive or negative. Common characteristics of all passive transport is that it doesn't use any energy and that it always moves from high concentration to low concentration. And that's it. Have a great day.